Good morning. Welcome to the second MSSC fall seminar. Today we have a double feature. We have some cool research going on in the intersection of computer science and bioinformatics. And we'll have two of our very talented professors talking about their research work. The first one is going to be by Chad Myers. Chad is an associate professor in the Department of Computer Science. He got his PhD from Princeton in computer science and integrative genomics departments. And he works a lot on using data mining techniques to learn about genome sequences and reason about diseases. Today he's going to talk about mining human genomes for genetic interaction underlying disease. Chad, take it away.
Okay, thanks for that introduction, and I'm happy to come and talk to you guys here. So before I start, I just wanted to say I'm happy to be interrupted. I have some slides. If I don't get through those, I'm actually glad about that. So don't feel like you can't stop me, and if there's something that's interesting or something you don't understand, I know that many of you probably don't have a really solid background in biology, so I'm happy to take the simplest. No question is too simple here. So if there's a side topic you want to explore, let's talk about it. So feel free to stop me at any point. Okay, so yes, I'll talk a little bit about, at a high level, some of the broad questions that our field of computational biology is working on. Just to give you a sense, so I want to cover a few of the basics of biology to make sure that not everything I say is totally flies over your head. So I'll make sure that we have at least a few of the standard things that we assume about what you know about biology. I want to start by painting a broad picture about what some of the big questions that our field is interested in pursuing right now. I'm going to spend most of my talk talking about sort of an example problem that we've been working on in my lab over the last decade, really, which involves trying to understand how genes come together to result in phenotypes, like the color of my hair, the color of my eyes, or whether or not I get a disease. So this is really a network problem, and so there's a lot of interesting computational methods that are required to make sense of that. And if we have time at the end, I'll give you sort of a sampling of some of the other problems we're working on, just so you have a sense for what types of data, what types of questions, what types of computational methods we're either applying or developing in the lab. So that's an overview of the talk. And as I said, I'm happy if we don't get through all of these bullet points. Okay, so first, just to establish sort of a very simple vocabulary. I'm not going to assume a lot of biology background, but there are a few things that are necessary. So first, we should start by saying that the basis of a lot of what's happening in biology these days is being able to sequence genomes. And genomes are composed of DNA. So this 4-letter alphabet that basically has the instructions. Think of it as the permanent instructions, or mostly permanent instructions, that tell all of our cells how to behave, whether they should form a certain type of tissue, whether we should respond to a certain type of stress. All of that's encoded in a genome that is composed of A, T, C's, and G's. And just to cover a few other basic topics, so that's stored in our genomes. That's the DNA stored in our genomes. What happens is parts of our genome, so our genome in humans is about 3 billion base pairs, so it's 3 billion of these A, T, C's, and G's. It's split up into separate units, which we call genes, which are on the order of a couple thousand, sometimes tens of thousands of base, sometimes a million base pairs. Genes can vary quite a bit in size. Those individual units are what we call expressed or transcribed. So that's a temporary copy of the permanent storage of information. And that transcription happens in response to environmental cues and in response to developmental cues. That's basically what actually makes cells do certain things, or make cells have certain morphology, or give cells certain functionality in our body. And by the way, all of our cells, for the most part, carry the same DNA. In my body, most of my cells carry exactly the same DNA. So what makes my heart different than my brain, different than my skin cells, is which of these units are turned on at different times. So which genes are expressed at different times. And this is what we call the messenger RNA. Messenger RNA is a temporary message that's actually translated by a machine called the ribosome in the cell into proteins. So proteins are really three-dimensional structures. They fold up. That structure is what provides all of the different functions in the cell. They form enzymes for the most part. They form enzymes. the thing that actually translates proteins itself is composed of proteins, right? So that's one example of machine. So proteins are sort of the workhorses, but the way they get there is through this process of transcription, and that's a context-dependent thing. So what I just described, most organisms have in common, sort of the central dogma of molecular biology. Okay, so everyone's okay with that basic? Yeah, go ahead.
This might be a little off topic, but how is the length of a gene determined?
How is it? That's a great question. So, do you mean from a species perspective or from a bioinformatics perspective?
You mentioned that like some genes are millions of base pairs in length, some are much shorter. How is that distinguished?
So basically, it relates to what is the ultimate. So gene sequence codes for protein structure, basically, which folds up. Some proteins can be functional with very small structures that don't have that many folds and are pretty small molecules. Others require a really huge protein just to be functional. So really that sequence varies based on what function that protein has evolved, what is necessary to actually pull off. And that does vary a lot, from a couple hundred base pairs even, all the way up to millions of base pairs. Some of the biggest genes in the genome actually are more than 1 million base pairs long. So you can imagine that's an interesting. Some of those genes that are really long are actually much more often associated with disease because they're big targets for getting problems with them, right? So you can imagine it's easier to make a mistake if you have to copy a bunch of 1 million base pairs versus 100. So that's sort of that length comes into play in terms of how often these genes are associated with disease. Good question. Any other questions about basic central dogma here? Okay, so one of the reasons that I'm a computational biologist is this plot right here, which shows you the cost of sequencing a human genome across the last decade and a half or so. And this line, I haven't updated this for a couple of years, but it's kind of flattened out. So basically, when they first sequenced the human genome back in about 2000, they came up with the first draft. That was a huge project. It cost about $3 billion. It was supported by labs across the world. So a huge effort, a huge amount of investment. That very quickly came down to in the hundreds of millions dollar range. Right after that first draft was finished, they got better at how to do this. And what happened in the next 10 years is really dramatic. So this is Moore's law. This is sort of the law that we talk about in computers all the time. The rate at which both the scale and the cost of this technology dropped precipitously starting about 2005 through 2012 or 13. So imagine the first human genome we ever sequenced costed $3 billion. Now we can sequence a genome for 1,000 or maybe a couple thousand, depending on what resolution you want. So that's a really dramatic, and just a matter of 10 years' time, right? So that's the time, just a little bit longer than it takes for a PhD student to train. We got from not even being able to tackle even one genome to right now we can sequence anything we want, right? So that shift really disrupts in a good way, the scientific field of biology. There's a whole bunch of things that we can now do in the last 10, even 5 years that we never really even thought, many of us, even technology-obsessed people thought were possible, right? So it's a pretty striking time. It's a pretty unique and interesting time to be involved in research in biology because of that. So just to give you a sense, this is sort of a timeline of sequencing. They sequenced the first genomes in the 90s, so the yeast genome. I'm talking a lot about yeast. Yeast is a commonly used model organism. By 2000, we had 4 or 5 different genome sequences, including the human genome draft. Zoom ahead, so mouse was sequenced shortly after that. Fly was finished about the same time. Zoom ahead to 2018, we have 40,000. I just checked this morning. We have 40,000 different genome assemblies, including viruses, bacteria, and there's tons of different bacterial species. And these are pretty small genomes, so they're relatively easy to sequence. But the bottom line is if someone cares about a species to any extent, it's valuable from a model standpoint, it's valuable from a medical standpoint, it's valuable from an industrial making beer, brewing beer, making bread, whatever, people have sequenced it. So this is not a bottleneck at all. If someone cares about a species, we can very precisely read out what its information code is in that genome. So if you take away nothing from my talk today, take away this, that even though we're really good at measuring genomes, and that's really accurate, so for the most part there's not that much noise in whether we get A, T, C's, and G's right. We can do that almost perfectly, with a few exceptions. we actually still don't really understand biological systems. So this is a picture of that. One way to capture that, now let's just focus on the human version of this plot. We can look at, so we know exactly the sequence of these genes. We can predict where the genes are. We know where they start and where they stop, and we can talk offline if you're interested in how that's done. So we know which of the units are. We know exactly what their sequences are. In some cases, we even know what the protein structure is. We know what it looks like if that thing is expressed and folds. But for about half of those genes, there isn't a single paper published in the literature about what it does. And what that means is we actually really don't know why that gene is in the genome. Is it helping to repair DNA damage? Is it responding to a certain kind of stress? Is it important for cell division? So these are the kind of functions that it'd be ideal if we knew what they were. So consider about half of the human genome, even though we know exactly what the part looks like in terms of the sequence, we actually really don't know how it contributes to human biology, which is pretty striking. If you think about it, we've actually had these sequences for more than 15 years now, right? So, and we still are making relatively little progress in just understanding how that whole system works. So it's like we have a very detailed parts list for a car down to the centimeter resolution, but we actually really don't even know, and we can look at the car, but we don't really know how all those parts fit into it, which is a pretty basic understanding of our species. And what I just described is for human, but if you even go back to the simplest model organisms, we work on yeast a lot. The problem's a little better, so you can tell yeast there's about a quarter of the genes. That's probably more like 15 or 20% these days. So we know more of what the genome does, but still it's pretty simplistic. So there's a lot of work left to be done. So I want to start just by sort of giving you that context and telling you a little bit about some of the broader questions. My lab isn't working on all these questions, but our field, as a computational biology field, we address a lot of different questions. So I want to give you a sense for that. So what are all the parts? That's sort of the sequencing exercise. We actually are really interested in, obviously, if there's a new species that someone wants to study, what are all the parts? What are the genes? We should sequence that. We should understand how it's different. What's their function? I just talked about this question quite a bit. When are they used? So that's a really important question. What makes my heart cells different than my skin cells is really about which genes are used at which times in development. And so that's a really sort of a critical way that we address that question is when are these parts used? A question that really is a center of most projects in my lab is how do all these parts, once we know the parts, we kind of know what they do, how do they interact to carry out processes? So as with most systems, man-made or natural, it's a complex system. There's many different pieces. We know what the pieces are, but the reason that life is adaptive, dynamic, robust is because all these parts work together in complex ways. And there's a huge community that essentially models cells as a network of pieces and trying to understand how the different parts interact to help carry out larger functions. Other questions are about how do genomes evolve. So yeast, half of its genes, or 6,000 genes, half of its genes we can tell have a really clear what we call homologue in human genome. So I can actually look at the sequence. It's not perfectly identical, but you can tell this gene derived from a common ancestor at some point. This is the same component, and we can look at that, and we can understand what was the process of evolution? How did it change that gene? What does that tell us about what kind of pressures were on different species at different time as these systems were evolving? So that's a common question that many people, what we call comparative genomics, comparing genomes. And the last one, which I'm going to talk a lot about today, is how do genomes vary across individuals? So how is my genome different than your genome? And how does that relate to things like my predisposition for certain diseases? So this is probably the biggest impact that genome sequencing technology is having right now, is that it's feasible now just not to sequence a single reference genome for a species, but it's easy to sequence all of our genomes. And understanding what makes us different is really the core of understanding how to treat medical conditions that are genetic, right? So that's a lot of work. And Professor Kwong's talk after mine is also addressing this question in a cancer context. So this last question is actually a lot of people are working on that in the computational biology community. And I should say computational computer science techniques, methods are really at the center of most of these questions. Okay, yeah.
Is there any research in building DNA from scratch.
Yeah.
Basically trying to figure out input output. How do you make a protein?
Yeah, so I would classify this, there's a whole field of what we call synthetic biology, and that's really the focus of synthetic biology is, actually it's a sort of a bottom-up way of understanding biological systems. We're building, we don't do this in my lab, but that field is building simple systems that, like they synthesize DNA or take parts from other systems and try to, for instance, can you construct an AND gate or an OR gate? Can you construct simple components that we understand and how we design systems? You know, the idea being if you construct a simple AND gate and a NOT gate, you have a full system to do whatever complex logic you want. Or I forget what the full complement is. You guys probably remember more than me. But yeah, so that's a very active field. And I think one thing that is enabling that on the industry side is it's gotten very cheap, economical to synthesize DNA sequences at a pretty big level. So I can come up with a gene and maybe a couple thousand base pairs long, even 10,000 base pair long, and I can order that and get it in 2 days for not that much money. A couple cents, maybe a cent a base pair or something like that, maybe half a cent a base pair. And that was not true even 3-4 years ago. So obviously the ability to synthesize specific sequences is really enabling for that synthetic biology effort because we can construct whatever pieces we want then, right? Okay, any other questions before we move on here? Yeah.
So since you're working with human genomes, like what is your source of data?
Yeah, it's a great question. So the scientific community, so this is largely supported and put in place by funding agencies. So the NIH, for instance, has a system they call the dbGaP. It's the database of genomes and phenotypes. And there's a lot of issues. So there's a lot of technical issues. You know, how do you store that much data? I would say that the harder part of that problem is the privacy issues and controlling access to that data, but providing access to the whole community. So dbGaP, if you want to learn more about that, just look up how the development of dbGaP. But basically, if a study is published that involves human genetic data, it's almost certain that and it's from the US, funded by someone in the US, it's almost certain that they had to deposit something in the dbGaP database. And when it's deposited in that database, what also happens is there's a data access committee that evaluates if I'm a researcher, I want to look at that. I want to use that data in some way. I have to go through a quite extensive series of tests. It takes, or not tests, but series of steps. What am I going to use the data for? Do I understand how to protect it? And so there's a lot of regulate. It takes me at least a month before even when I know the data's there, until I can get the data, which is a bit of a hassle. But you know, we're protecting. The challenge here is if someone's genome is deposited, we can figure out who exactly that person is, right? And there's been papers on this that from a genome, you know, there's enough ancestry and other information that's available now. You can actually fairly easily for some people figure out exactly who they are. So, and that's going to only increase. So there's a lot of thinking about how to protect people's privacy, but also make the community able to look at this data. But yeah, dbGaP is a good thing to look at if you're interested in how that happens.
Yeah, this might be a little off topic, but I've heard that like DNA has like a 512-year half-life, and so something like that,
yeah,
where it breaks down like so the idea of being able to recreate a dinosaur, say, from amber is probably never going to happen. But but I did hear that there are some like dormant genes and stuff in today's birds.
Yeah.
Do you see any of that happening from like?
So there actually is pretty good. So I don't know the context for that state. I actually don't know what the half-life of DNA is, but it probably is 512 in some environment. But I do know that they've sequenced Neanderthals' genomes, right? So if it's preserved in certain ways, amber or I don't know. Actually, I don't know that field very well, but if it's preserved in certain ways, they can sequence. They've sequenced Neanderthals. They've sequenced what's the elephant, the woolly mammoth, right? So that genome's been sequenced, and actually there's a lab that's actually making a woolly mammoth right now. So George Church's lab. If you're interested in this kind of thing, you should look up George Church's lab. He's one of these crazy visionary guys. He's developed a lot of the genome sequencing technology. He has really crazy ideas. You know, maybe 10% of them work, but they're a really big deal. And they're actually making a woolly mammoth right now. So and I think the way their approach is to take an elephant's genome, and they understand what the differences are because they were able to sequence a woolly mammoth's genome, and they're essentially changing the elephant's genome in all the places where they're different. And they're gonna use an elephant, I think, to carry that embryo. And I haven't seen the latest progress, but I wouldn't be surprised if they have the first woolly mammoth in the next couple of years. Maybe, I haven't followed that project, but it's pretty fascinating. So we're talking genomes that are tens of thousands of years old, for sure, hundreds of thousands of years old. I don't know what the oldest one, but 10 to hundreds of thousands of years, they've been able to sequence a genome if it's preserved properly. That's, of course, not easy. It has to be exactly the right circumstance, but it's happened. Yeah.
For dbGaP, do a lot of these DNA testing services also feed data into that database, or are they too coarse?
No, they will, and they won't do that because that's their business model, is to protect that. So I'm going to talk a little bit about 23andMe in a second. So 23andMe is probably the most successful of the consumer DNA testing so far. I think they've got a couple million people banked. I actually don't know what they have now. I knew a couple years ago they passed 1 million. But their business model is that it's their data. And and by the way, when you buy it, so I've done this, and a lot of my family have done it. It's about $130. They don't sequence your genome, but they essentially profile it. I'll talk more about that in a second. But so their model is they were going to do this 10 years collecting data. Now they're cashing in on it. So now they're going to drug companies and saying, okay, we've got 10,000 people in our database that have Parkinson's disease. You're working on Parkinson's. We can give you that population, work with you on that population to figure out what genetic factors are in that. So they would not have a business without protecting their data. And yeah, everyone that's been profiled through those sites has signed away their rights to that data. So when you there's a big agreement, you're agreeing that they own it. I actually don't know. It's actually, it scares me a little bit. I don't know what they can do in terms of, can they sell it to an insurance company that's providing me insurance? I actually don't know what they're allowed to do. I haven't read the agreement that close. I probably should have. But I think this is actually the ethics and the regulations around all of those things are lagging behind the technology in a lot of ways. And that's a big issue. I think there's a lot of interesting questions, important questions there. Yeah.
So without cooperation with those private companies, how would you identify somebody from, or was it dbGaP profile?
Yeah. Oh, how would, yeah. So that's a good question. So if you send me email, I'll point you to, there's been a couple of papers that show an example of this. They got access to genetic material, and they were able to pinpoint where, the exact person or where they lived or something. But it's because of, so there's these sites like Ancestry.com. It's really what those companies share to other people, including 23andMe. So in 23andMe, I get told when there are people that are related to me in that database. So if you take someone's genome and are able to query a database, you get family names associated. Most of those, like at 23andMe, they tell me what the family names of people are that are related to me because people are interested. That's my third cousin that's in the database, and I know their family name. So from family names, other information, you can then start to make inferences about where geographically this person is, and pretty soon you can actually intersect that with all the public records, and you've got the person, right? So and there's a paper that kind of goes through that process just to illustrate the potential problems with privacy here. Yeah, question.
So it's kind of like the same way that they found the Golden State Killer.
Yeah, it's similar. I think it's exactly that kind of process. Yep. No, that's a, and I forget what the details of that case were, but it's similar. Yeah. Did they use Ancestry.com? Is that right?
No, it was another one that I hadn't heard of. But it
was something. I think it was like in the same class as Ancestry.com, but it was a different.
Yeah, law enforcement has managed to get access to a lot of that.
Yeah, and that's another question. Does 23andMe have to give my profile to the law enforcement? I don't know actually what. Can the law enforcement just say, hey, I know you have a profile for Chad Myers. Can you give us his profile? Because we need to match it against something else. Obviously, I'm not committing crimes every day that I really care about that, but it's a valid question. Yeah, question in the back there. Yeah.
Just trying to get relevance in our program to yours. So I know, downtown, there's a national marrow donor program, And they use Hadoop.
I work there.
Yeah.
Oh, you work there? Oh, okay.
I'm one of their software
Yeah.
engineers.
Okay, so you guys use Hadoop and do a predictive. So is this
Yep,
a
and
type
we're on
of?
AWS, and we use it for mainly our diversity goal, which we refer to diversity as less than three or less people on our matching site that have a match. So we're looking for people that have only one person out there that they can match with, or maybe two, and we're working from an 8 out of 8 match of the genome to, with the new drugs that are coming out, a 6 out of 8 or sometimes even less than that to avoid the graft versus host disease and that kind of thing. So we use a lot of AWS and that kind of stuff.
So your question is generally about what are the tools that enable these kind of things? Is that?
Yeah, it's just trying to understand
Yeah.
what, you know.
Yeah, I think cloud computing, big, because a lot of these problems are, you're running things on, 1,000 or 10,000 genomes over and over. You have to do this process. So a lot of these, it's a matter of scale more than it is problem complexity for a lot of these things. Not to say there aren't hard problems in biology, but so resources allow you to scale things up and accomplish them pretty quickly are important. So in the clinical setting, when people get genomes, they don't want to wait 10 days for you to assemble it and call which the variants are. They want the answer in an hour, right? So, and these are important for clinical decisions. They're starting to use that. So a lot of the scaling issues are to support actionable decisions quickly from genomic information. There's a lot of interesting computational infrastructure challenges there. My lab doesn't work a lot on that, but there are a lot of academic labs that are working on these questions of privacy. How do you transmit genome information? How do you scale to these kinds of resources? There are a lot of academic labs working on those. I just don't happen to be one of them, but that doesn't say they're important issues and they are being addressed at multiple levels of practicality. Yeah. Okay, so maybe just one comment. I wanted to give you a sense, like the flavor of our work. And you probably will get that sense from some of the specific projects I talk about in a second, but it's highly collaborative. So, and this varies a little bit based on the style of the lab, but in my lab, we work on problems that typically involve at least 2 or 3 other labs that are more experimental than us, right? So, of course, there's tons of public data out there, and you can get access to it, as I just said, but a lot of times the most interesting questions require you to sort of team up with someone that really has expertise in a specific biology domain area and maybe is generating unique data. And then you can really start to do interesting things that push the envelope of our knowledge of biology or biomedicine forward. So a lot of the problems relate to managing and interpreting large multidimensional data sets. So that means machine learning, data mining, statistical methods in general are the basis of many of our approaches. So and it's not always developing novel algorithms in that space. Sometimes it is when it's required. Sometimes it's using the tool sets that exist in a smart way for the right problem. And that's something that, again, kind of varies based on the lab. Some people are attracted to problems that require novel algorithms. Others, just want to have impact. And there's a lot of impact to be had by applying approaches that are pretty well worked out in the computer science community to new areas that people have never used those approaches before. I would say at least my lab's work is tied to or motivated by biological, biomedical questions. So that's not true of all bioinformatics people. I think other people maybe are more motivated by how interesting the computational problem. I think that varies quite a bit. In my lab, we sort of work on problems that, if I were to pose the question, the biology and biomedical community would say, yeah, that's an interesting question. If you can solve it, we're interested. So. There's tons of those questions, so you can work on a variety of problems. We do. You'll see it. You'll get a sense for that. So we work on a variety of problems in my lab. So to give you a sense, my lab's interests and expertise are really about network analysis and algorithms for network data in a biology context. And I think that for me, the really interesting thing is that we can work on a variety of different systems and questions with a common toolkit. So we can be experts in a certain computational area, but that can actually impinge on a whole bunch of different questions in terms of biology. And that's different. Probably some of you know of people that work in biology, and what often happens is you have to specialize. So you have to pick, in order to get to a point you can actually do something interesting, you might have to invest several years of your life learning how to be a yeast geneticist. You have to learn how to grow yeast in the lab. How do you manipulate their genomes? How do you do all the various techniques that are sort of organism specific. If you want to do mouse geneticists, same thing. You have to learn how to do all the practical things of raising and manipulating mice. That's changing now because with genomics, the toolkit is pretty common. So I think even in the biology side, that's changing quite a bit. People aren't as pegged as a single organism biologist. But in computational biology, it's always been true that we can be generalists in the sense we can work on problems across the whole domain. And yet we can still use a common toolkit like how do you make sense of networks? And that can apply to a lot of questions. So we work on things from trying to understand cancer better or develop drugs for cancer to why do different varieties of maize look different or have different properties in the field, to basic biology in organisms like yeast or C. elegans, which is a worm system. So, and all of these are come from a common toolkit of networks and analysis or inference of networks. And I kind of talked about this. The kinds of techniques that we either develop or apply. So data mining, so unsupervised clustering is a huge thing that we do. Collect data, try to see where the structure is, what happens, what is that structure related to in terms of questions we're interested in. Association rule mining is a class of methods we use quite a bit to try to derive interesting specific structure in data. Even classic approaches like PCA, unsupervised analysis of data, actually get used quite often. A lot of problems we approach from a predictive standpoint. So can we build models that predict something interesting? Like can we, from your genome, predict whether you're susceptible to a certain disease? Or from a cancer genome, predict which drug is potentially the best to treat an individual with? So that obviously suggests machine learning methods would be relevant, and we do apply and develop a lot of different machine learning methods. And that's a huge area of development right now, especially with the deep learning techniques, infrastructure in place, and really having a lot of success in a lot of different domains. There's a lot of activity in this space. So in terms of machine learning applied to biology data. And I would say graph algorithms. So we work on networks, so a lot of the classic, so CS has a rich history of analyzing, developing algorithms for graphs, and many of those approaches apply in our setting and are quite useful for specific biological questions. Okay. So I want to transition into talking about a specific project, and the motivation here is related to genome sequencing. So just to give you some perspective, the thing that, so we talked about genomes being very cheap to sequence now, but what happened was in 2009 we had about 3 genomes sequenced. So 2000 there was a single reference draft of the human genome. And that was a pool of Caucasian males, basically. They used as reference genome. In 2009, the technology had gotten cheap enough. They actually sequenced Jim Watson, one of the discoverers of the structure of DNA. He got his genome sequenced as sort of a PR activity. In 2009, that costed about $1 million. But that curve started to go up. By 2010, we already had a couple thousand. 2011, there were pretty good statistics. There were something like 30,000 genomes sequenced. Now, I actually should have updated. I'm sure there are several millions of individuals' genomes sequenced, and maybe at varying levels of resolution, right? So this is not always, at the depth that we would do for the reference human genome sequence, but there's hundreds of thousands, millions of human genomes, either tumor genomes, normal genomes, different types of tissues that people have looked at that exist. Many of those are accessible by the public through proper access methods. But that's a big deal. So just to give you a sense for that, so there's been a lot of progress made. And one of the things that's been done are what we call genome-wide association studies. So I sort of alluded to this a second ago. And this is a fairly simple idea. The idea is if you could collect a couple thousand people's genomes that have a certain disease, one thing that you can imagine would be interesting is if you can take those individuals and contrast them with otherwise matched controls. So other people that same ancestry, other factors you might same age, things like that, that don't have the disease, and then you profile their genomes and you ask what's different in the people that have the disease at a single place in the genome, as compared to people that don't have the disease. So I can say, is this mutation, does it seem to be associated with the occurrence of this specific disease? So that's a really simple idea. And the idea is you wouldn't just do that for one locus, but you do that one place in the genome. You do that across every place in the genome that you looked at. And that's why we call it genome-wide. And association is the process of checking for this enrichment that I just talked about. So that has been repeated for hundreds of human traits, human diseases, not just diseases, but also traits like hair color, things like that are more complex. And dbGaP contains the results of those studies. So there's more than 5,000 studies, and this plot shows you between 2006, where we had very few of these associations, in 2018. So let me just play this movie. So this is the human genome. So different chromosomes of the human genome all lined up here. So we have 26 different chromosomes. But each one of these dots that's emerging here is a specific place in the genome, a single mutation was associated with a human trait. So most of those are disease traits. And so that's just between 2006 and 2018. So in 2006, we had relatively little knowledge of these things, a few places here and there. In 2018, that picture looks like this. So this is just from May. So what it means is we've learned a lot about, okay, this place in the genome is associated with a person's risk of getting Parkinson's or a person's risk of having gallstone complications, whatever the disease they were focused on is. We actually have learned quite a bit about a set of different loci. And you might find, by the way, for Parkinson's, it might be 20 different loci. There's one here, there's one over there, there's one over there. So these typically are spread throughout the genome. And there's typically multiple factors that come up for any given disease you look at. So it's not like it's a single gene that causes it. There are 15 or maybe 20, maybe even 100 different loci that seem to influence people's risk. So that's actually it's a lot of progress. There was a lot of money invested in this by NIH and other funding agencies. That's a lot of progress, but actually the really disappointing thing from that is, I'll talk about in a second, and I'm going to use my genome as an example. So I actually have my whole genome sequenced. It's on this hard drive. And I had it, I don't know, 4 years ago already, so it's actually been quite some time. There was a program that University of Minnesota had the sequencing company come and they paid for it. So it costed about $5,000 then. I probably wouldn't have paid for it myself for $5,000, but just to give you a sense for the data, on that hard drive there's 1.4 billion reads, we call them. That's basically short segments that are 100 base pairs each. That's how sequencing technology works. It can't give you huge stretches that might compose a whole chromosome. It gives you short stretches that are like 100 base pairs at a time, but a lot of them. And what's done is basically you align those to a reference genome, and where they mismatch. And you usually have so much depth that you align for any given place in the genome. You have many reads that overlap with that. So you can imagine this process of taking each of these reads, figuring out where it goes based on the reference, lining them all up, and you might have 20 reads stacked at a given base. If all of them agree that there's an A here and not a T is in the reference, then we call that's a difference. So that's at a high level how these approaches call variation, right? So I have about 3.4 million places in my genome that are different from the reference, which is a Caucasian genome, right? So its background is my general ancestry, but I still have 3.4 million places where my genome is different. And this is pretty typical. So about 1 in 1,000 bases varies in all of us if you take two individuals that come from roughly the same ancestry. Yeah, question.
So can your genome mutate? And if so, if you got resequenced today, could there be differences? And would that tell you anything?
That's a great question. So our genomes mutate all the time. Every cell division, I forget the number, it's between 1 and 5 places in the genome will randomly get replicated incorrectly. It's not many. If you think about it, there's 3 billion different bases. So the process is pretty good, but when you replicate a cell, you get about one error per replication event. So the question is, how does that affect us? So the cells that form an embryo, the sperm and egg cells that form an embryo, if those get mutated, that's passed on to the next generation, right? If I get a mutation, I get mutations all the time. If I go out in the sun, I get mutations in my skin cells all over the place, right? And that's if I expose myself to, I had a beer last night, I exposed myself to a mutagen. So some cells, probably in my stomach, other places, did get a mutation. Those aren't going to affect my offspring unless they happen to affect what we call a germline cell, right? That's not something that's passed on, but those are things that cause cancer, right? So all that you need is one cell to get a mutation that affects a process that, for instance, stops cells from growing. That gene no longer works. That cell doesn't know how to stop growing, grows out of control, and takes over your whole system, basically. So does that make sense? So we get mutations all the time. Obviously, we try to control our exposure so we get less of them, because the more you get, the more you expose yourself. If you smoke, you're getting a lot of exposure in your lungs, basically. And prolonged exposure, you're going to get a mutation in a place that causes cancer at some point. That is basically the effect of accumulating mutations. Does that make sense? So my cells across my body are different, and that's an interesting question now, because they've been exposed, because when you divide, you pick up mutations. But mostly they're the same. And where they're different, those are the kind of things that result in cancer in us. So the question up there and a question over there. Yeah.
So aside from the genetic variation in an individual's body, how much has the error rate in terms of the sequencing process been looked at?
Yeah, it's been analyzed a lot. That's obviously, in order to be confident in this data, we have to know what the error rates are. And you can imagine, if you do the math here, there's 140 billion base pairs of sequence. If I take 1.4 and multiply it by 100, because each of these is 100 base pairs, that's 140 billion base pairs worth of sequence, which means every point on average, I have, what is 140 divided by 3? It's too early for this kind of math. Like 50, I have an average 50 reads that stack up at every point in that data. So, and you need, in order to be very confident in the sequencing, you need that kind of coverage to be confident that it wasn't just I got 10 that happened to have an error in the same place. And that's especially true because sequencing machines make errors non-randomly, which means they can overlap and be wrong and agree on a wrong base, and that's a problem. So the depth has to be high enough to sort of account for that error rate. So and that is something that is the main criteria that sequencing technology companies are focused on, is what is the error rate? Because we want to tell, I only have 1 in 1,000 bases that's different. If my error rate is 5%, that's way above the kind of thing we're trying to measure. So that's a really key, and it's been analyzed a lot, and that's one of the main drivers of that whole industry is what's your error rate. It's two things. How long is your read, and what's your error rate? The longer the read, the more you cover at once. The error rate, the fewer you need to be confident about something. There was a bunch of other questions, I think, before. So up there, yeah.
Yeah, it's a similar error rate question, but rather than in the technology, rather than in the machines, more about I guess the sample set, right? So if you're looking for a certain disease and you sample, like if I take cancer patients and I sequence their DNA and try to look where that happened, like how do I know I didn't get the one cell that was mutated? Is it just I cover a whole bunch of different people to try and have?
Yeah. So it's a good question. So it's about how you sample. So like when I got my genome sequenced, they took a lot of blood. And so they're sequencing not just one cell, they're sequencing a population of cells. So I'm sure in that population I had cells that were different because of the errors that I just talked about. But what you get is sort of a consensus sequence. And that's a very confident sequence of what I inherited basically from my parents. It's a really good question. And one of the new really exciting technologies that's happening right now is called single cell sequencing. You can actually sequence individual cells' genomes, which is pretty crazy. You can take a population of cells, sort them one by one, and get a genome sequence just for that cell, So we're actually getting a lot more information about how is this cell's genome different than this other cell. And there's variation there. As you know, this idea that I just said that all of our genomes are the same across our whole body is not actually true. It's mostly true, but there's a lot of interesting places where you get variation even beyond these random errors that I just talked about. So. But yeah, it's most of this, what we call bulk sequencing, is you do a population of cells, so you really get a sort of average across many cells. There was a bunch of other questions. Yeah, go ahead.
Is the cost staying kind of consistent at about $5K right now?
I think it's, for the kind of technology or the kind of approach I just talked about where you're resequencing, like you have a reference and you just want to call variants, it's more like 1,000 now. So even that was 5 years ago or 4 years ago, maybe. It was 5. So that's already come down to it. I don't think it's going much below 1,000. I mean, it's maybe 500 for companies.
What's driving that cost down?
So I could give a whole talk about that. There was a dramatic shift in how these things were sequenced, and it was about doing. And there's a lot of interesting chemistry behind it. So if you're interested in chemistry and that question, it's a really good read. So you should read about a company called Illumina. That's where I got mine. That's really a company that drove a lot of this change in sequencing technology. It was really about parallelizing how you get reads and making the reagents not so expensive for that. So essentially, what used to happen is you get one read every couple of hours or something like that, or it was maybe, I don't know what it was, 67 bases an hour, I think something like that was. I forget what the old tech machines do. Now you get 100 million reads over a couple of days, and it's because they parallelize the whole process. So that's a really interesting read just from a chemistry industrial technology. There's a lot of interesting innovation there. Okay, I think there were a couple other questions. Yeah, go ahead.
You said earlier that you're pinpointing different diseases and being able to understand where that nice graph that you have with a lot of dots on it. Any person can have multiple morbidities at any point in time.
Yeah.
How do you know which one is which?
You mean for a given person?
For a given person.
Yeah. You're getting at it. That's a really tricky question. It's really a question of when you form a case group for one of these studies, the disease group, are you actually sure that that person has the same disease as the other person? Or do they have a different form of the disease, or they have that disease because they had this other condition? And most of us do probably have multiple conditions at the same time, and some people are taking medications that interfere with different things in different ways, right? So what that gets at is when we form these case control groups, it's not as clean as that whole design suggests, right? You probably actually have 100 different diseases represented in that population. That changes based on some diseases are highly homogeneous, meaning that the individuals they present with exactly the same symptoms. Other diseases, so autism is a good example of that. There's kind of a characteristic autism phenotype, but there are a lot of different ways that that's manifest, and we still classify that as, and I think we've gotten better. Classification systems for autism specifically, there's been a lot of work on that. We view that as a spectrum. We view that as potentially a cluster of different things. But that's what's happened there is not untrue of other phenotypes, that it's messy. And yet we try to cut through that mess and make a case control because the statistics demand something like that. And that's sort of, yeah. Ideally, you would do one of those on each of the individual sub-diseases, and then you really get to understand the clean genetics of that specific disease. And you probably have this intuition, but one thing that's important for these studies is having a lot of statistical power, which is all related to how many people you have. So there's a trade-off between collecting people that have exactly the same phenotypes and just collecting enough people that you can even do something interesting. So that's an important trade-off that's struck there. Yeah.
Sometimes you're in the news about designer babies.
Yeah.
And for example, they'd be like, oh, okay, I want to make a baby like J-Lo or something.
Yeah.
Get her genes and make a baby. So how do they do that?
Well, first, it's possible to do that, but the ethics of that are a huge issue, and so no one's really doing that right now. It's completely possible to do that right now. We'll talk a little bit. Most of you have heard about CRISPR technology. This is a technology we're using a lot in my lab and with collaborators. You can edit genomes actually surprisingly easy now, and that was never true before. So we'll talk a little bit more about CRISPR in a bit. But so that's not really happening. You can't design designer babies right now. People are selecting embryos for certain properties. I think the closest thing to that that's happening right now, there are a few companies that have developed around the family planning space. So you can get your potential partner's genome sequenced, your genome sequenced, and they're providing recommendations about really important things like, are you going to potentially have kids that are highly susceptible for a certain disease, like cystic fibrosis? If you're both carriers, that's important to know that because you have a 1 in 4 chance of a kid having cystic fibrosis, and that's a life-changing condition. It's good to know that in advance before before you decide to have kids or whatever, right? So that space is getting pretty sophisticated. It's sort of the genome consultation space about partners, and they say, here's your traits, here are places where you're likely to potentially have issues. You know, you could probably say your kids, I'm sure those, I haven't done this myself, so I don't really know. I'm sure those companies do things like your kid's likely to have blonde hair, blue eyes. I don't know. I actually don't know what they do, but that's probably part of their business model, even if they don't really publicly talk about that. So there's a lot of ethics in that space, right? I mean, because the lines, the technology, it's possible to do things, but the ethics are a totally different question, and it's a sticky area. And I don't, like I said, I think the ethics have lagged the technology, which is probably true in most areas of technology. I mean, probably make the same argument about social network technology, right? There's a lot of debate right now about how that's harming us, right, as a society, as a country. Anyway, so are there other questions at this point? This is good. So we're having a lot of, I like this kind of discussion. Okay, so let me just say, so I've got 3 million bases that are different. I'm not going to talk a lot about why this number is so low. So most of our genome doesn't actually code for genes. Genes are pretty, I forget the number. It's about 5 to 10% of our genome, maybe even less, is gene space, which means there's a bunch of sequence in between that is, we know it's important. We know some of the things it does, but it doesn't directly code for proteins. So of these 3.4 million, 10,000 actually affect a protein. So there are 10,000 ways that my proteins that are expressed are different. That is, they fold differently. It affects how well I see, how, what my chance of going bald, things like that, right? Those are things we know it's affecting the so that's a lot. That's actually a lot of differences that could contribute to disease, other things. So when you get this, when you get a genome sequence at this level, you go through a process where you see a genetic counselor, and so I did that before I got it sequenced because they make sure that you know what you're getting into. And when you get your results, you talk about that with a counselor, and there's a sheet that says clinically significant findings. And that sheet is what I have on this slide. That's it. So after my genome was sequenced, I talked to this counselor. There was one thing that they call clinically significant in my whole well, let's say the 10,000 that affect a protein. And it turns out there's a gene AMPT1 that it's known to be associated with a muscle disorder. This basically affects how well you recover from heavy exercise. What's known about it is you need 2 copies of that bad version of that gene in order to have that disease. I only have 1 copy, and as far as I can tell, I don't really have any issues with muscle recovery. So I don't have that phenotype. It's something that's significant. If my wife happens to have that same variant, some of our kids might have that phenotype, obviously. My point is that that sheet is really short compared to how much data I collected about my genome. And that is highly typical. And at the same time, I should say, I know that my family is susceptible to a bunch of different diseases, right? So my mom has had 3 different types of cancer. I had an aunt die from cancer last year. So there's a history of cancer in my family, heart disease. There's a bunch of problems that I can tell, diseases that travel in my family that I'm sure are encoded somehow in our genome. And yet, and we have it 50x coverage. I'm sure that is high quality. And yet, we can't really predict a lot of these things I know I should be able to predict about my genome. Yeah.
Not to be morbid, but you had it sequenced 5 years ago?
Yeah.
Have you updated the list since?
Yeah, so that's a great question. So in fact, I haven't had time to update the list, but we are learning a lot, right? So this list gets longer every day as we make progress, as those dots accumulate on that network, so I haven't. Yeah, as soon as I find some time, I'll actually do that. It's one of those things, like, for most interesting questions that I have, I get graduate students or undergrads that are interested in projects to work on that. For this question, it's kind of weird because it's about my genome, right? So it's like,
I'm not saying like you need to tell us everything that.
No. But I think it's a good idea. It's strange that a person that's interested in data, I haven't had time to do that. And for when I have questions about genomic data, I usually work with students to do it. This is one thing I don't feel comfortable doing that because it's like, it's a conflict of interest. It's like, work on this data to figure stuff out about me. Not that I care about the privacy side of it. It's more like I don't feel comfortable having someone. And it's interesting. There's actually if you want to read about, there's actually a professor I know pretty well. This guy, Mike Snyder, is at Stanford. He has a whole bunch of papers on what he calls personalized, I don't know, personalized omics. But he tracks everything about himself. I don't know if he's still doing it, but he did it for several years, not just his genome, but every day he gave various samples, and he had a whole part of his lab working on analyzing that, right? And there's a lot of interesting papers. Like he tracked what happened right after he got some kind of virus, and he was in a pre-diabetic state, and it looked like he was potentially transitioning to a diabetic state. So there's a lot of interesting things and he changed his lifestyle to avoid that. So it's sort of a story about what you could potentially do if you're instrumented and track yourself on a daily basis. But if I were him, I don't think I would have done that, just because it's kind of weird to have a bunch of postdocs and students analyzing data that's collected from yourself. Anyway, that's a side fact. But it's a great question, and that list will be longer today, I guarantee you. Yeah.
So with this data, you're really looking at specific bases and sequences and how that directly affects a trait or protein. Do we know a lot about how the one set of sequences or one set of bases might affect the others, if they're sharing data back and forth and interacting in non-direct ways or correlations?
I guess you're talking about if this mutation and this mutation together cause an issue, is that right? Or
something like that. So
that's exactly what my lab works on. So almost all of this, all that research, if I showed you all those dots, this list of clinically significant findings is all based on this paradigm of take this sequence, is that individually, is that associated with disease or not? And as I described in the beginning, these systems are complex. We should definitely expect there are going to be cases where by itself it doesn't mean anything, but in the context of some other change, or in the context of an environment, let's say I expose myself to certain things and I have this bad gene, now I've got a problem, right? So that's exactly what we work on, is trying to be more to actually test complex combinations, and that problem is even harder than the individual one, because if I've got 3.4 million places I'm different, 3.4 million choose 2 is a huge number, right? So the statistics of that problem are really challenging, but we think we have some interesting ways to tackle that. So that's what we work on.
I was kind of correlating it to what I do, which is completely different, but it's infrastructure as code, whereas you have a recipe or a sequence of logic, which in some cases accepts instructions, in other cases checks the environment, and in certain cases it will replicate something and spin up multiple copies of individual pieces of infrastructure, and how that that kind of sequencing correlates to kind of this. I was just wondering how.
Yeah, no, it's definitely affecting whether or not we have disease. It's been a problem that's just too hard to tackle. We're trying to make a little bit of progress in that area, and it makes perfect sense to ask that. I think there was a question over, yeah.
So since this analysis is done by looking at the differences between your genome and kind of the baseline genome, have people looked at the baseline genome to see what they're susceptible for?
Yeah. So that there's been a lot of improvement. So there's a lot of issues with this idea of a single reference genome. First, those people might themselves be strange in different ways, and you can tell that, or just have rare diseases that not everyone should have, and you can tell that as soon as you start sequencing a couple thousand individuals, you can see, oh, that variant that's in the reference genome is actually pretty rare in the population, and maybe they have a specific disease because of it. So that's one thing that has improved a lot. There's a lot of efforts just to characterize what is natural human variation across the globe, really. So the other big issue, which is not talked about a lot in the scientific community, but it's a big problem, is many of the studies I talked about, genome-wide association, you have to collect a population. So how do you get a population? You go to a hospital, you get people to sign up for the study, and that process and access to patients really biases what types of people, what ancestries of people are involved in those studies. So a large fraction of GWAS studies are based on Caucasian genotypes. And that means that there's a lot of stuff that we haven't discovered, and there's a lot of different variants in different populations. So it means that a lot of what we discovered from those studies isn't relevant to people of other ancestries. So now GWAS studies are being done more and more on diverse populations. So people are characterizing genome sequences across the globe. They're going across the whole world sampling what are the different genetic variants that different populations have, and the idea being that you can actually form a reference for each one of those individuals in a certain area. Imagine sort of going out and finding a reference that's relevant for your ancestry, and then based on that reference, what mutations might you have that might cause disease? So that's a great question. Let me just talk about a couple of things. I can tell you a little bit about what we found based on 23andMe data for me, but the message is all the same. So how many of you guys have had 23andMe done? How many people have had? Okay, so actually it's a pretty fair number of you. So I think the experience varies quite a bit. So I did it before the FDA shut them down in terms of disease risk. Nowadays there's a lot fewer diseases that they report on, but it's increasing every year. So basically what happened is they originally gave you information about a bunch of different disease risk up front. The FDA said, okay, this is not okay. We have to get every single one of these reports approved for a specific disease before we release that to people. So other traits like whether I flush when I drink alcohol, bitter taste things, whether I'm going to go bald or not, these are viewed as not disease related, and so they're not restricted. And so these are still available to everyone, and disease traits are becoming approved every year by 23andMe. So anyway, you can get a sense here of some of the traits that you can try to predict based on your genome. There's some interesting things like increased odds of baldness. I'm approaching 40, so that's probably going to happen soon. Anyway, you can get a sense. Some of these are pretty clearly right. Others, even the easy ones, seem to be wrong. There are some interesting. I'm not going to talk about this, but this is a pretty exciting finding for me. There's a muscle performance category, and it says I'm a likely sprinter, which is news to me. It turns out there's maybe the quick version of the story. There's a gene that is associated with fast twitch muscle fibers. And my mom and dad were sequenced. By the way, you can clearly tell who your parents are when you have this done, or whether your parents are actually your parents. So this is maybe the most significant, I think one of the most significant results of this 23andMe. There are surprises along that. Like people learn, oh, you're not actually my parents, and that's not something they knew, which is a life-changing thing. That's not a trivial thing, right? So anyway, so it turns like you can tell from this, you can tell from all the loci that in fact my mom and dad are my mom and dad. In this case, my mom gave me a C, my dad gave me a T. So my mom really should have been a sprinter. I had some potential. So the C version of this gene is the good one for sprinting, the fast twitch muscles. My dad didn't have any potential, but neither my mom.
What you're saying is it's really lucky your dad caught your mom.
That's probably right. That's right. That's good. I'll steal that joke from you next time you just talk. Yeah. I'm not creative enough to think about that. That's good. Yeah. Anyway, so this is just a fun story about sprinting. But more seriously, if you look at, so I'm able to see all of the disease risk, and it's interesting. So anyone that had things done before a date that FDA shut them down still can access that, and they're now giving people access to diseases as they get them approved.
Sorry, on that first page it said risk for malaria.
Oh, is that right?
Yeah.
Malaria resistance.
Yeah, malaria resistance.
Malaria resistance. That's right. That's right. Should not. Yeah.
Well, it's not a fun disease, but if you had gotten malaria, is there a possibility that could change?
Not based on the kind of. So this is a. So first, you can tell by other means, by antigens in your blood, if you've had malaria or not. But based on this test, this is DNA that I inherited. And as I said, it's averaged across a bunch of cells, saliva cells in this case. And so this would not reflect my exposure in my lifetime. There are ways to check that, not through the saliva you donate to 23andMe. That's a good question. Okay, so the disease, back to the other diseases. So this is pretty typical. So I'm showing you this not because I have sort of a clean record, but because it's modest predictions. So these are the worst case scenarios for me in terms of elevated risk. I haven't done this for maybe a year or so. I should probably go back and check. Again, same questions before. But the worst case for me is gallstone complications. The background risk in my matched population, Caucasians, is 7%. So that's sort of if you average across that population, 7% of us are going to have complications for gallstones in a lifetime. My risk is 11%. That's the worst case for me in terms of enrichment, right? So Statistically, that's a big effect. If you look at a population, it's a 1.6x increase in risk. That's the kind of things that these studies find. But at an individual level, 11% versus 7% means almost nothing. I guarantee I can push that number a lot higher by my lifestyle. I haven't looked into that, but there are ways I'm sure that I can cause gallstone complications, and it's going to increase my risk a lot more than those genetics. So, and the lesson's the same, that this doesn't mean I'm not at risk for things because of my genetics. It means that we just don't understand how to interpret it. And it relates to this question we had before, that the rules are probably more complicated than just looking, do you have this variant or not? And if you do, it's associated with this phenotype. And that has become pretty real across many traits, and it's been described as a problem called missing heritability. And what that means is that we can tell diseases are genetic. We can measure that. So you can actually do twin studies that twins were born and separated at birth. You look at to what extent they get the same traits, same diseases. There are other ways to estimate how much of a given trait is genetic. And what's found in most diseases, if you put all of the loci that we found, that huge plot that I showed you before, all the dots, you put them all together, and you use that to predict who has the disease. In most cases, you explain way less than half of the genetic portion of the disease. So, and these are some numbers. This is a little bit old now, but actually not much out of date. So the best case scenario is macular degeneration. We explain about 50% of what we should be able to. Again, this is of the genetic part of the disease. We can tell it travels in families. We can still only explain 50% of that. For many diseases, it's more like 10 or 15 or 20%. So it's usually a lot even less fraction than that. That reflects that we just can't find it, right? So we can find loci that are associated. We put all those together. We still can't predict. We can't predict disease that well. And so this gets back to the question that was asked. There's a lot of potential reasons for this. There's a lot of caveats to these studies that have been done. One big caveat I should tell you about is that most of them focused on variation that was at about 5% frequency or higher in the population. So that means they looked at 1,000 people, and if there weren't at least 5% of those people that had a variant, they didn't even study it. And that's partly for statistical reasons. You wouldn't have power anyway to say something about the variants that were rarer than that. But that's obviously one thing. That's something that people have looked at in other diseases. Go and look at the really rare. What's the variant that has 0.1 frequency in the population? Maybe that's the reason that people have disease. Another one that we focused on, this is related to this question, is maybe it's not the simple model of this single variant causes disease is overly simple. Maybe it's that it's combinations. Maybe it's this combination or this variant here on chromosome 1 and a different variant over there, one of which I might have inherited from my mom, one from my dad, or maybe I got both of them from my mom. Maybe that's why you're susceptible to a disease. So this combination idea is something that we explore in my lab a lot. It's important to communicate how hard this problem is just fundamentally. Because the problem is basically moving from this model of finding a single variant and testing whether it associates with the disease. I think we can all understand that problem. But imagine doing that in most of these studies, we focus on either 500 or 1 million loci, so about the number of variants we expect in a given genome. But imagine trying to test, does a person have, find all the people that have both this variant A and the second variant X, and now do that for all possible variants that you see in the population. So that's take a million choose 2 combinations. That's something like 500 billion possible tests that you'd be looking at. Is this combination associated? Is that combination associated? Is that combination associated? Computationally, actually, we could potentially solve that problem. It's not a computational issue. It's a statistical issue. So when you do that many tests, some of you might know, you have to dial up the level of stringency at which things have to pass the test in order to have confidence. It's called a multiple hypothesis testing issue. And what that means is we actually don't have enough people for some of these diseases to have the power to find things if you do it in this way. So, I only have a couple of minutes left, which is great. You guys have had a lot of good questions. But I want to just paint a really quick picture of how we're tackling the problem in the lab. So what we've done over the last 10 years since I started my lab here, we've worked a lot on a way simpler version of this problem based on this model organism, yeast. So Saccharomyces cerevisiae, I mentioned this before, It's got 6,000 genes, and it's easily manipulable, which means I can easily knock out genes, or my experimental collaborators can easily knock out genes, knock out combinations of genes, and we can study how it affects these cells. And this is work that's been going on for a long time, but the idea basically is to use, I'm just going to show you a picture here, to use a robotic technology to construct many, many double mutants. So the concept is we take a clean genome that we know exactly what everything is, we introduce 2 changes, and only those 2 changes, and then we measure what happens. And we do this across the whole genome, right? So we do this essentially exhaustively so that we can essentially measure how often these kind of interactions happen, when they happen. Do they happen between certain types of genes? Do they happen at a certain frequency? Does that network have certain structures? And so that is basically what we've done in collaboration with this lab at University of Toronto. They have really done a lot to develop the experimental technology to make that possible. We've done a lot of the developing methods to process that data, interpret that data, and learn things about it. And I'm going to just give you sort of a very short summary of what we did there. But the really striking, by the way, we've knocked out about 23 million double mutants in that organism. So there's 23 million different pairs that we've knocked out and measured what happens in that context, which is something that until very recently, like the last couple of years, you couldn't do in really any other species. So that's something that only that model could allow us to do. And we found something really striking, which is when you find, so first, there are many interesting cases where you knock out a gene, introduce a mutation, it has no phenotype at all. Knock out a second gene, it also has no phenotype by itself, but when you knock those out together, you see a really strong phenotype, like yeast cells die. We find about 500,000 cases of that in yeast based on the 23 million pairs that we looked at. Yeah, question?
What are you measuring?
We're measuring, you see those pictures of plates that I just had up? Those are about that big. It's each one of those dots that you saw is a colony of yeast that has a very specific genotype, and it's grown up over 2 days. So we're measuring how fast does it grow. Something actually very simple that you don't even need a microscope for. We take images of that and we segment them, and we essentially measure the area of that colony after 2 days of growth. And what's pretty interesting is if you can precisely knock things out, measure even a crude phenotype like that, you can actually learn a lot about how the genes are related to each other just because you're introducing specific combinations at a huge scale. So that's something that I'm sort of framing it in a disease context, but maybe I should show you this. We're sort of running out of time, but I want to show you this because this is important aside from the disease problem. So this is actually something that we've learned. So each of these dots here is a gene, and what I'm showing you is not the interactions that I just described where you knock 2 of them out and you have a surprising phenotype. These are what we do, and pretty much like everything in data science these days, it's about figuring out who's similar to who, right? So you can construct a matrix of these interactions, and actually the most interpretable thing you can do with it, which maybe is not that surprising, is to look at who shares friends in that network. So which genes have the same patterns of interactions with other genes, and where you find that, generally they are related to the same process or involved in a very similar process. So think of this as like a social network of the yeast genome, which genes tend to be closely related to each other. And what's really interesting is that if you look, so this is totally based on our data, but if you look inside these they're very highly dense clusters, these are clusters of genes that seem to all look the same in terms of who they interact with. They're highly enriched for genes that we know to be involved in a specific process. There's 80% of the genes in this cluster are involved in DNA replication repair. These clusters are involved in vesicle-mediated transport. So the idea is that you can actually take a species we've never seen before. If you're able to in combination, manipulate genes and measure even a crude phenotype, like how fast do cells grow after that, you can sort of reconstruct how everything is related in the cell. And this is a really powerful concept. So just to put this into context a little bit, with CRISPR technology, you guys have probably heard about CRISPR in the news. So CRISPR basically is an approach that was it's a natural system that bacteria use to fend off viruses. But a number of scientists basically leverage that system in the context, and now it can be done in any cell type really, but in human cells it's being used quite frequently. You can essentially target certain areas of the genome for mutations. So while it wasn't possible to do these kind of things in human cells until recently, you can now introduce combination of mutations in human cells fairly easily. We've already done about 1 million combinations in human cells so far. It's following exactly the same kind of thing we did in yeast. The concept is we want to build a map like that for the human genome so we know how all the genes are functionally related. So like I told you at the beginning, about half of those we have no idea what they do. If I can tell you who they're related to, that gives me a really good start on what functions they have, because often we know, in the case of human, we know about what half of them do. If I can put other, this is what Amazon does. This is what all these companies, these database companies do. They profile you, you look at who you're similar to, and then they sell the same products to you. We can play the same game here. We can profile genes, look at who they're similar to that we know what they do, and we can label them and say, hey, I think this gene has that function. So that's basically a sort of picture of the lab. We've learned a lot about how genes interact. And maybe just to wrap up in 1 minute here, the most striking thing we learned relative to this disease question is that when you find interactions, I'm just going to show you one plot here. When you find genetic interactions, they tend to be highly structured in yeast. So this is actually a picture of human, but the idea is the same. Basically, you rarely find an interaction by itself, like two genes together cause a phenotype and they're off by themselves in a network. Typically, they exhibit this really specific structure, which is a complete bipartite graph. And that means that I can knock out any gene over here and combine it with any gene over here, and I see the same extreme phenotype. So what it says is these networks have a very specific characteristic structure. It's highly non-random. If I use that same structure when I go and tackle the human problem, I can now find these interactions. So I'm not testing individual pairs because that's 500 billion pairs and I can't do it statistically. I can test for bigger structures like that. And the reason I know to test for them is because we did all this work in model organisms to do it. So this sort of gives you a sense of the type of work we approach, sort of the network side of things. I'm gonna stop now just to give you guys a little break. Maybe let me just acknowledge a few people. There's a lot of, I haven't talked a lot about the stuff in the lab, but there's a lot of people that help make this happen. So the kind of approach I'm talking about, this interaction question, was really driven by two students. Wen was a former PhD student in my lab. Gang was a PhD student that was in Vipin's lab here, Vipin Kumar at the University of Minnesota. He's now a professor at Mount Sinai in New York. And there's a whole bunch of people that support this broader yeast study that I sort of mentioned at the end here. Many of these are PhD students or postdocs in my lab. We work a lot with other scientists. So Toronto is a major source of collaboration. Charlie Boone and Brenda Andrews' lab. So I think that the last thing I want to say is that a lot of these projects really require academic scientists to work in diverse teams. I think you guys, most of you probably are in industry, I think, right? I think industry has been way better at that, probably than academia. The academy has, just because we haven't had the incentive to do that. People can work in their own office and solve their own problems, and that's generally worked okay. Science is much harder now that you actually sort of need to work across these teams. So a lot of what we do requires contribution from a lot of different groups. So with that, I'd be happy to, I need to take off after this, so I'm not going to be able to stay around for Ray's talk. But I'm happy to talk to you guys offline. So send me email. Happy to meet up if you guys have questions. Yeah, feel free to reach out and happy to talk more if you're interested. Thanks a lot. There's a lot of interesting questions.